Dear colleagues, I would like to present the result of the study Direct Optical Generation of Singlet Oxygen Induces the Apoptosis of Tumor Cells. This study was realized on the basis of cell physiology and pathology laboratory of Research and Development Center of Biomedical Photonics of Zarel State University. As is known, biological tissues need a constant supply of oxygen to ensure the processes of vital activity. 90% of the oxygen consumed is accounted for by mitochondria to produce ATP. In addition to generating energy, mitochondria are the main producers of reactive oxygen species. At low concentrations, reactive oxygen species can be involved in multiple regulations of physiological and biological processes. The accumulation of reactive oxygen species as a result of any imbalance in their production and metabolism causes oxidative stress, which leads to damage to cellular components and to the development of serious diseases. The accumulated experience in the study of physiological and pathological role of reactive oxygen species and identifying the mechanism of their generation form an interest in the singlet form of oxygen. This form is characterized by great reactivity, unlike the triplet form of oxygen. Today, there are two mechanisms for generating singlet oxygen. The first mechanism is the use of photosynthesizer. It has wide application in medicine during photodynamic therapy, but this approach is characterized by a number of disadvantages, in particular, direct cytotoxicity of photosynthesizers, uncontrolled localization of photosynthesizer in cells, and inducing cellular stress not only of tumor, but also of healthy cells. The second mechanism consists of the direct excitation of oxygen molecule by light at specific wavelength. By now, there are more and more papers that study this approach for its use in the biology and medicine. However, the main disadvantage it is low absorption of light. It can be compensated by light intensity or exposure time. In addition, there are different in the observed reactions, and taking this into account, we assume that singlet oxygen can affect the viability of different cells in different ways. To study the effect of singlet oxygen on cell viability, a developed device was used. The device includes a laser of 1267 nanometers to excite the oxygen molecule and the control laser of 1122 nanometers. The control laser was used to confirm the relationship of recorded processes with the generated singlet oxygen. The dose of laser radiation was 200 joule per square centimeters. All studies were conducted on cells of melanoma and on cells of melanoma and control cells fibroblast. The measurements were carried out by the confocal microscope. You can see the experimental setup and how the laser radiation for singlet oxygen generation and control laser radiation were supplied during this study. At the first stage, using the singlet oxygen sensor green, which is highly selective with respect to singlet oxygen, it was found that exposure to 1267 nanometers laser as the chosen dose activates the production of singlet oxygen in both cell types. At the same time, 1267 nanometers laser does not cause the production of other reactive oxygen species, in particular superoxide, hydrogen peroxide, and does not affect the production of reactive oxygen species by mitochondria. Thus, the 1267 nanometers laser at the chosen dose selectively produce only singlet oxygen, which does not change the enzymatic production of other reactive oxygen species in fibroblasts and melanoma cells. Although in our experiments, the 1267 nanometers laser did not affect the formation of superoxide and hydrogen peroxide, singlet oxygen can also cause oxidative stress. measurement of the main endogenous antioxidant glutathione, which is sensitive to redox changes and reflects oxidative stress, show that produced singlet oxygen does not change the level of the main antioxidant. But analysis of the mitochondrial membrane potential revealed various reactions in the study's cells. The changes of the mitochondrial membrane potential were observed in melanoma cells. The shape, could you please, okay, video. The shape of these changes was different. Fast and complete loss of membrane potential or its oscillation were associated with the opening of the mitochondrial permeability transition pore. Pre-incubation of melanoma cells with an inhibitor of mitochondrial permeability transition pore, cyclosporine, prevented the loss of the membrane potentials. Since the opening of the mitochondrial permeability transition pore is involved in the mechanisms of necrosis apoptosis, using special fluorescent probe, we estimated the percentage of cells in these conditions 24 hours after laser exposure. And it was found that generated singlet oxygen does not cause the necrosis, but it activates the apoptosis in melanoma cells, and the percentage of apoptotic cells after laser exposure of 1267 nanometers laser increased by 10 times compared to cells without exposure, and cells after exposure by control laser for fibroblast with reaction was not observed. And the main conclusion is identify specific vulnerability of melanoma cells to singlet oxygen compared to control cells of fibroblasts may be used as a potential treatment of tumor in the medicine. This work was supported by grant of mega grant and grant President of Russia Federation. Thank you for your attention.